Hi, it's Mrs. Vance again, and this is the review for the Unit 9 test. What's on the test? There are, as usual, 40 multiple choice questions. 9 of those questions are review questions over things like DNA and RNA, protein synthesis, the cell cycle, all of which are relevant to what we're learning about now with chromosomes and meiosis. There also is a bonus question worth 6 points. It has pictures where you have to identify events in the phases of meiosis, and you can get partial credit for getting some of them right and not all of them. So do your best to study, look at various pictures that you can find either in the notes or worksheets or online to see how the various stages or phases of meiosis are illustrated so that you'll be better prepared for that. So, as a review, meiosis, remember, is the reduction of diploid cells to haploid. This is how we get gametes, or sex cells, that have half the number of chromosomes, which is important to maintain the chromosome number of the species. In the whole process, there is one replication of the DNA in the S phase. There is an interphase that precedes the entire meiosis process and has an S phase where the DNA is replicated, but that's followed by 2 separate divisions. Meiosis 1 separates the homologous chromosomes from each other. Remember, the tetrads join up, the duplicated homologous chromosomes line up together in the center of the cell, and then the homologous chromosomes separate from each other, and then meiosis 2 separates the chromatids from each other so that we get 4 haploid daughter cells as a result. Okay? Crossing over is something that's really important that happens in meiosis. It happens every time in meiosis, and this is where homologous chromosomes exchange portions of their chromatids with each other, which mixes up genes from the mother and the father. This occurs in prophase 1, and remember, this happens in prophase 1. The duplicated homologous chromosomes line up, forming that tetrad that's made of 4 chromatids, and then while it's in the tetrad formation, then portions of chromatids trade places, and that mixes up the genes. It usually happens in multiple places at the same time, and those the places where it occurs are called chiasma, or chiasmata is the plural. Chiasma is a single one. I don't believe the term chiasma is on the test, but I wanted to remind you of that. The final results that we get from meiosis, that we have one replication of the DNA. We have 2 divisions, meiosis 1 and meiosis 2. They have the same stages as mitosis, prophase, metaphase, anaphase, telophase, and it produces eventually 4 haploid daughter cells, which are genetically unique from each other because of crossing over and because of the independent assortment of the chromosomes. I like this table really well, which compares mitosis and meiosis so that you can see the same, what happens in the different ones, how many divisions, how they compare to each other, that you have diploid cells or haploid cells, and so forth. And so this is a really nice summary chart that summarizes the similarities and differences between mitosis and meiosis. It's really easy to get confused here, so pay close attention to the big differences there. All right. Here's a diagram also that shows a comparison of mitosis and meiosis, showing what happens with the chromosomes and how the crossing over occurs there in meiosis 1, the formation of the tetrad and the chiasma there, and how the homologous pairs separate from each other at anaphase 1. And then meiosis 2 just is involved in separating the chromatids like you see, much more like you see in mitosis. Sometimes there are mistakes that happen, and one of the mistakes that can happen is something called non-disjunction, and this is when the homologous chromosomes fail to separate properly. This can produce gametes with extra chromosomes or fewer chromosomes. And if the faulty gametes are fertilized, this leads to genetic disorders of various kinds, generally trisomies and monosomies, which generally cause some kinds of problems for people. I would recommend, as you're studying, that you go back and look at the various videos. There's a video in today's folder. There was one in another folder that was an Amoeba Sisters video about chromosomes and karyotypes. There was another video from the Amoeba Sisters about meiosis. And you can look on YouTube as well. And of course, you can always go back and look at the notes videos. But looking over those things will help you recall what's going on in these processes. This is not a real difficult unit. It's just a little bit confusing sometimes because of the similarities with mitosis and the differences from that. And so just study for your test. You should be able to do well on it. The test is tomorrow. Make sure that you are logging in sometime between 8 o'clock and 4 o'clock. Make sure that you're able to submit your test before 4 o'clock, because otherwise it may time out on you and you'll end up with an incomplete grade. Do your best, and we'll see you next time. So long.